Welcome, everybody. So for the 3 p.m. track, it is the how to grow a Mars base. Sustaining life on Mars is more than just whether a plant will grow. So may I present to you Morgan Irons, founder and chief scientist of Deep Space Ecology.
Hello, everyone. As was said, my name is Morgan Irons. I am a student at Duke University and the founder and chief science officer of Deep Space Ecology. I am currently working on my double thesis in environmental science and biology. My biological research consists of testing of pretreatments for the growth and survival of crops under Martian regolith conditions, for which I started writing my experimental procedure in September of 2015 and commenced preparation and testing in January of 2016. Here's some pictures. Although I have learned many interesting things about what to do and what not to do, and though this information is vital to being prepared and equipped to grow plants on Mars, I can tell you that sustaining life on Mars is about more than whether a plant will grow. My talk today is not on my current biological experiments, but is on my environmental science thesis that I started writing and preparing for in January of 2015 on the development of closed ecological systems, for sustainable space habitation. Today, I am going to address what my environmental research indicates are requirements for establishing a permanent human presence on Mars. So to begin, what is a closed ecological system? An ecological system is a system involving the interactions between a community of living things in a particular area and its involvement with the non-living environment. A closed ecological system, or CES, is an ecosystem that is sealed off from everything outside of it. You can imagine that this is very important in space, considering humans and plants cannot survive in a vacuum. Living on Mars, a CES would provide a separate biosphere for living things such as plants, the microorganisms that would help the plants grow, the humans that need the plants to thrive, and the air, soil, water, and energy that support the cycles of life. Much of what is known today about closed ecological systems for use in space is based on research done by the Soviet Union beginning in the 1960s. However, the idea of developing a CES for space habitation was written about in 1895 when Konstantin Tsiolkovsky published his book, Dreams of the Earth and Sky, in which he depicts and details a CES on a space station. In the US during the 1960s, NASA was first considering the possibility of using bioregenerative systems in life support, but it wouldn't be until 1978 that they initiated their Controlled Ecological Life Support System program. In conjunction, a group of mainly ecologists known as the Botkin Group met to determine how ecology would play a role in bioregenerative and life support systems. Partnering with NASA, they came up with 3 foundational elements for developing a space-based CES. 1, the life support system would resemble a terrestrial farm. 2, humans would be a major component. And 3, the purpose would be to support human life. NASA would go on to use these foundational elements as a basis for future approaches. However, not everyone was thinking along these same lines. Biosphere 2 decided to utilize entire biomes in its CES design. It incorporated 7 different biotypes in one structure, those biotypes being tropical forest, ocean, desert, steppe, field and farm, and the human habitat for a crew of 8 persons. The first experiment was in 1991. The Biosphere 2 experiments received criticism for their endeavors to recreate Earth in a closed structure. Biosphere 2 ended up having to be opened due to the system experiencing ecological malfunctions, such as environmental disruption driven by the unnatural adjacency of the 7 biomes, technological disruption from the curing cement of the enclosure absorbing oxygen from the atmosphere, biological disruption due to decomposition in excess of growth and ratios not found in natural development of flora systems, and psychological disruption of humans due to the stresses of a maladjusted ecological system. What we learned from Biosphere 2 is that setting up a self-sustaining ecosystem is about more than putting a bunch of plants together in a dome. And yet, Biosphere 2 experiments seem to try to embrace the concept that Russian researcher Kamyshlov worked to popularize. Quote, "The stability of the biosphere as a whole and its ability to evolve depend on the fact that it is a system of relatively independent biogeoconoses which compete for habitat, substance, and energy, and so provides for the evolution of the biosphere as a whole." Since Biosphere 2, many more efforts have commenced to enable humans to live in isolation from their daily Earth habitat while meeting the space mission criteria. Human models have been specifically designed to test systems close to external human interaction. While helpful in predicting the mental and physiological effects of long-term isolation, those models are conducted within the natural environment of Earth and are not meant to test ecological feasibility. Human technology models test the ability of engineered systems to replace the functions of Earth's natural ecosystems. These systems work well when there are regular resupply runs and a means for humans to get back to civilization quickly in the event of a technological failure or accident. For example, the International Space Station receives regular resupply shipments from Earth to replace parts, food stores, air scrubber chemical packs, water purification filters and consumables, and to haul away trash and spent cartridges. The ISS also has the Soyuz capsules that can be used by the crew to escape quickly if necessary and be back on the surface of Earth within hours. Human technology biology models introduce bioregenerative technology in place of man-made technology in an attempt to reduce the need for man-made consumables and repair parts. This particular model considers the need to reduce the cost of the supply chain to remote areas like Mars. It helps make human space exploration more politically palatable. These new models also start using concepts like in situ resource utilization and in situ repair and fabrication to reduce the cost of prefabricating parts on Earth and shipping them to remote locations such as Mars. The general idea is to ship just the right selection of technology and biology to Mars in order to establish an Earth-independent mission. NASA's definition of Earth independence is 1,100 days. But can human tech biomodels truly accomplish this goal? Well, let's see. The schematic on the slide is from a patent that was issued for an ISRU plan. It is designed to support a Martian greenhouse and/or hydroponic system for growing food. It is a typical engineered system meeting standard engineering principles of function and performance, redundancy as needed to mitigate the risk of downtime, and do so at minimum life cycle cost. It eliminates the need for some spare parts by incorporating bioregenerative technology, thus reducing Earth to Mars supply chain costs. I would bring your attention to the portion of the system that uses microalgae to produce oxygen. Consider such a system operating on a Mars base. All systems are up and running, and we have been growing food for a while. We are near the end of our initial 1,100-day run, at which point we will get a resupply of some spare parts from Earth. Suddenly, microalgae bioreactor line 1 goes down. No problem. This is a critical system, so we have a redundant line 2 that we can bring up and operate while troubleshooting and fixing line 1. Then, line 2 goes down before we have even started troubleshooting the problems with line 1. Given time to investigate what went wrong, we would discover that all of the microalgae died in both lines. What killed the algae? Did the epigenetics or genetics evolve to a non-functional state in the alien environment of lower gravity and higher ambient radiation? Is there an alien biovector present in the Martian environment that we had not discovered yet and that killed the algae? Maybe a trace chemical in the Martian water. This kind of problem takes years for biologists to figure out, but our Martian explorers don't have years. They have to make a decision now on whether to do a forced evacuation. So, what is the real problem with this thought experiment? Is it that we did not know about this adverse effect on the Martian environment on the microalgae? Or is it that we did not have sufficient resilience to adapt to a problem that we did not predict or know about? I suggest that answer 2 is the more productive answer to consider. After all, we will never know everything we would like to know before we put humans on Mars. So, how do we make a human stay on Mars more sustainable? In other words, with no need to do a forced emergency evacuation. To understand this, we need to briefly review the history of human adaptation and social development. Earliest archaeological records have humans in small groups of hunter-gatherers living in a locality in which they have become familiar with the local natural resources. From there, humans adapted and evolved to an agrarian society in which they had a central support village and surrounding crop fields. Modern cities are what eventually came from this adapted process. It can be said that one of the reasons for human survival is their ability to adapt quickly to unplanned events. The tribe of humans living in a cave ran out of local resources due to overuse. They need to find more resources, so where do they find them? They find them out in the wilderness outside of their known locality. Later in history, a local village is hit by a blight against their main food crop. What do? They go into the wilderness to find the food resources they need to survive until they can get to their next harvest. Recently, when New York City metropolitan area was hit by the Hurricane Sandy event, supplies and resources came from beyond the immediate agricultural zone surrounding the city. The point is that we cannot establish a human ecological system that will actually support the survival of humans without something that is called competitive redundancy. So the problem with the bioregenerative system in our thought experiment is that it was designed with supportive redundancy in mind. Indeed, this is the most efficient engineered system one could build on Earth, a non-alien environment that has all of the resources and spare parts relatively nearby. But nature reveals to us that supportive redundancy can lead to system failure or degradation. There are natural ecosystems on Earth that function in a supportive redundant system, having evolved in isolated locations undisturbed by alien outside influences. These natural systems have components that are the only things that fill their respective niches. They have no competitors. They are either the only provider of a resource or they are the only consumer of a resource. However, if a disturbance occurs to such a system to weaken or kill a component that is the only thing that fills its niche, the entire ecosystem can collapse. Thus, it is key to have a resilient system that has multiple living organisms competing for resources and multiple supplying the same resource. This way, if a particular organism dies, competitors can take up their market share, thus preventing the ecosystem from collapsing. In summary, all past and existing attempts at designing systems that will permanently sustain life in space with minimum to no support from Earth are at high risk of failing unless these two weaknesses are resolved. I propose a model that seeks to solve these two weaknesses. The three-zone model is based upon the way humans have always lived on Earth. As I described earlier, consider a city. An urban ecological center not only has the city as a human habitation zone, but also has an agricultural zone within and outside of it, and a buffer zone of wild nature that encompasses both the city and the agricultural areas. The habitation zone is needed to provide humans with a safe and healthy place to live and work. The agricultural zone provides for a place where humans can grow the food they need. The ecological buffer zone provides ecological services needed to keep the habitation zone and agricultural zone healthy, while also providing a biodiversity that is needed for competitive redundancy. I don't have the time to go into the landscape theory and ecological principles behind the importance of an ecological buffer zone, but I can tell you that it provides movement of energy and nutrient cycles throughout the system, where without it, the system would become stagnant, as in Biosphere 2. Consider for a moment a mission to Mars that builds following the same model. The mission uses modules with fully engineered ecosystems as the habitation zones. The mission plans to have 30 crops in its agricultural system that will be planted in both hydroponic and soil systems. The mission also has over 1,000 different seeds for plants that grow on Earth in dry and cool climates. We build the structures, set up the zones, harvest the in-situ resources from the Martian environment, plant the agriculture in a planned fashion, and plant the 1,000 plus species of ecological buffer zone plants in a scattered fashion. Over the course of attempting to grow the crops, we discovered that even though all 30 of our crops had success on Earth, 20 out of 30 of them fail on Mars. It is likely that we could have such a miserable success rate due to the fact that the plants are growing in an environment that is alien to their previous evolutionary path. What will likely cause the most challenge is that the microbiology that we bring with us that is needed to support the growing of plants will evolve quickly in the alien Martian environment, and some will change the way they function, adversely impacting the plants and humans. So what will our humans do? They will need to adapt. They will need to go out into the wilderness of the ecological buffer zone, find the plants that are succeeding in the alien environment, and domesticate them into the agricultural zone. The plants that are failing will be moved out into the ecological buffer zone, where they will be allowed to grow wild and compete. Thus, the third aspect of the model that raises the chance of success for our permanent Mars settlement is providing our humans with enough resources to adapt to an alien environment. As the plants that we take and the humans that stay on Mars continue to live and adapt on Mars, they will evolve into a uniquely Martian ecological system through ecological succession. My company, Deep Space Ecology, has been working on the designs for a CES that meets the criteria of the three-zone model. The Mars Epic X1 design is based on the standard circular dome model. Its dimensions of 170 meters in diameter are the minimum that we believe to be necessary to support a pace with 8 humans while mitigating the risk of failure of settlement continuance. The dome area contains both the agricultural zone and the ecological buffer zone, laid out in a pattern that maximizes the distribution of the ecological services to the areas used for agriculture and for habitation. The dome is transparent and provides no gamma radiation shielding. We believe it to be vital to allow the plants to adapt and evolve under the Martian ambient radiation conditions so that they become a uniquely Martian ecology. The habitation zone is placed outside of the dome area. It is comprised of standard modules delivered from Earth to Mars with standard ECLIS systems. After connecting all the modules together, they will be buried in 2 meters of regolith to provide radiation shielding and insulation to the inhabitants. What is true about this entire setup is that things will not go as initially planned. Thus, the design intent is to enable human adaptation and survival in an alien environment with minimum to no support from Earth. Deep Space Ecology has already commenced developing our Mars Epic X2 design that allows for establishment of a human foothold on Mars and the gradual buildup of a CES to the requirements developed under the Mars Epic X1 design. We are working on preliminary plans with Astrolabs with intent to build the Mars Epic X2 prototype starting next year. The prototype would not only test the final configuration, but would also test everything required to build it and to grow the ecological system in an environment simulating Martian conditions. Individuals who wish to contribute can do so starting in October through our crowdfund partner, Orbit Muse. Individuals and companies wishing to collaborate can contact our CEO, Lee Irons, at leeirons@deepspaceecology.com. Now, I have something cool to show you at the end of the presentation, but first I need to make some acknowledgements. I couldn't have started down this path without the help of Duke University and my advisors at Duke University. The biological research I am conducting under this grant that Duke University provided me will be peer-reviewed and made publicly available through publications. It is also available through my research Facebook page. I would also like to give a thank you to JPL scientists William Abbey and Gregory Peters for advice and collaboration, and Sire Industries for donating the Mars regolith simulant I am currently using. I'd also like to acknowledge the wonderful people I have met through social media, especially Tessa McAvoy, cartoonist at large, Dr. Robert Zubrin, whose book inspired me, as I'm sure most in this room it has, and who personally encouraged me at the Humans to Mars Summit last spring to submit my abstract for today. And Abby and Nicole at the Mars Generation, where my upcoming research blog geared towards teenagers who wish to enter the space sciences will be posted. I'd also like to thank everyone who helped make up the critical difference in funding my university research. More funds are needed if anyone would like to donate. Gotta put that personal plug in there. On the business side, here is our incredible team at Deep Space Ecology that is working on the CES designs. You can read the bios of our team members at our website, www.deepspaceecology.com. We are also still interviewing for various positions. Interested individuals can submit their information to our onboarding team. In closing, I would like to show a video of our future state concept Mars Epic X3 design, which works on reducing the size of the footprint of the ecological system by moving towards a vertical architecture. Thank you. Okay. Okay.
Very good. I absolutely loved it. Let's go to this gentleman here.
Hi. You have the in the dome. You have the buffer zone and I guess it's the managed plants that your agriculture zone all together under the same dome.
Yes.
Can you explain how they will behave differently, or what's the difference between the buffer zone and the main zone there?
Okay, so yeah, that goes into the landscape theory and ecological principles of how the ecological buffer zone helps with this. So what we've seen in past research is that if you just have an agricultural manhandled zone in a closed system, it will eventually fail because of degradation of the nutrients in the soil or the soil itself, things like that. What the ecological buffer zone does, it acts as a natural wilderness. So if you were to go out to a farm, it's open to the air, birds, animals can go in and out of it. There's usually trees surrounding it as well, and other ecological systems. So we're pretty much basing it off of what you would see here on Earth. And having that natural ecological buffer zone allows for movement in the system. So nutrients and services that are being produced in the ecological buffer zone will be able to move into the agricultural zone and vice versa. So it creates that movement in the system that you would find here in ecosystems on Earth.
I love the fact that you included the ecology. That's the first presentation I've seen that detailed. How do you plan on warming the water? Are you going to use the water in the soil already?
Yeah, so we're doing more research on how exactly to do that. And we have a technology engineering team that's looking into that. I know a previous presentation had the drip irrigation, which is a great idea with creating a sustainable water source. With the biological research that I'm doing, determining different pre-treatments for the regolith, I'm finding that you don't necessarily have to have completely sterilized water. I'm using non-potable water to water my plants because to see if you would need to fully sterilize it and how that would affect the plants.
You mentioned a lot about the flora. Do you have any comments about the fauna need for this? I imagine there'd be an extensive environment of insects, like bees and things to pollinate things, and it might be an ecosystem a little bit larger than what's going on here. Do you have any comments on how to introduce something either primitive or something smaller for this sort of scale of ecosystem?
Yeah, definitely. So currently, the plants that we are selecting and looking at don't require pollination by insects. They're just wind pollinated or self-pollinating. But when it comes to introducing insects, we're looking into that as well. I'm good friends with the director of the research greenhouses at Duke University, and he recommended using beetles. I can't remember the specific species of beetles, but these beetles can pretty much live in extreme environments, and then you can also eat them, so they can also be a food source. So yeah, choosing the different insects and looking at the research that has been done on the International Space Station, that will help determine what plants that we decide to bring to Mars. Oh.
Hi. So for your prototype that you're describing, is there any planned specific location? Like, are you trying to match insulation levels similar to that of Mars? Like, say, maybe go to like where the F-Mars location is, or are you trying to go for like the most convenient location possible to build it?
So with our collaboration, oh, well, do you want to? Okay, so the CEO of my company is here, and he can give you more information about that, actually. Lee Irons, who's also my dad.
My camera to our social media manager. We've brought Skidge into our team. Some of you might know her. She's currently the Hera 12 mission commander at NASA Johnson Space Center. She's a PhD geologist, planetologist, and she is one of her tasks is to study both the existing sites that have been identified by various parties and government agencies, and also look at some of our own sites based upon our own criteria, based upon in situ resources available, based upon the layout of the land, whether we can take advantage of certain geographies and such. And so that's something we're working on. For a loom ecologist and an entomologist. So if anybody is one of those, let us know.
One is possible to be able to model and simulate on Earth issues, of course, with things like radiation and 40% gravity. Which do you think are the most serious impediments or risks in terms of how this works on Mars? The question is, if you plan to use Martian soil, what are your thoughts on what you have to do with it to make it good food for human or Earth plants, given there's perchlorites and other things that probably most of our plants wouldn't like very much?
Definitely. Okay, thank you. So to answer your first question, I would say the gravity. I'm more worried about the gravity than the radiation, because we have examples where there are major events like Chernobyl, and microorganisms growing in the environment, even if it's not good for people to be there. So with the problem with gravity is very important because that helps pull the air into the soil, helps pull water into the soil. And if you have a lower gravity, it's not going to be doing that as much, and that could be harmful to plants because you don't want them to suffocate. So currently, we don't have really any that besides the space station, but that would be one of the things I think we need more research about, if we need like a suction pumping system or not. Yep. So we're working on the perchlorate problem. There are certain microorganisms that are present here on Earth that can be used to break down the perchlorate into less harmful chemicals, and we use that in systems that have been damaged by chemical spillover and other such things, where they're actually treating the area using certain microorganisms. So we're looking into that for that. When it comes to making the soil more habitable for plants, that's what my biological research is on, determining those different pre-treatments, whether they need solid as fertilizer, what kind of microorganisms they would need, having an organic litter layer versus not having one, using a water treatment, or going based off a completely dried system.
All right.
Okay.
So first off, I really enjoyed bringing ecology into the Mars discussion as well. So I see we're using Mars regolith. Is there any plans to test Mars air composition, see how plants will grow differently?
Mm-hmm. Yeah, so one of the experiments that I'm planning to do hopefully in the next year or so is working with hypobaric chambers and testing the increased CEO concentrations. And there's actually research that you can use to already get a foundation for that, and that's having to deal with increased CO2 concentrations with global warming. So there's a professor at Duke University that I had, and her research is specialized just in that, how the plants would be able to react to increased CO2 levels. And there's mixed results. It really depends on the organism and the species that is being tested, but there tends to be at least small to a little bit higher CO2 levels that actually improve plant growth. Okay, thank you so much.